Hello, this is Mr. Wartsky again. This is the third screencast in the energy series. This is a slide of the mitochondrion that you should have drawn in your notebook. And now with all the different parts to it. If you haven't gotten it before, pause it and let's see if we can get it down. So we have finished the glycolysis. We have talked about the mitochondria. We are on to the next step, which is called oxidative decarboxylation, or ox-decarbox, or short, if you're friends of oxidative decarboxylation, that's what you get to call it. Pyruvate enters the matrix through facilitated diffusion. Pyruvate has charge, there's cell membranes involved, can't get through the hydrophobic middle layer, needs to have a protein tunnel. That's old hat. So pyruvate, we're going to take 2 electrons away from pyruvate and a hydrogen ion. We know that a dehydrogenase has to do that, and it will add a molecule of coenzyme A to pyruvate, and now it's called acetyl-CoA, and believe it or not, a carbon dioxide is lost from that molecule. So now acetyl-CoA has 2 carbons instead of the 3 carbons that pyruvate had. The enzyme is pyruvate dehydrogenase. Coenzyme A, interestingly enough, is formed from the vitamin B. I don't know why it's not called coenzyme B or why it's not called vitamin A, but vitamin B, that's why it is the energy vitamin. It gives you a structure called coenzyme A, which allows you to continue through aerobic respiration. Now, acetyl-CoA will enter the Krebs cycle. The Krebs cycle is also called the TCA cycle. It's called the citric acid cycle. It's called all these different cycle names. It was discovered by Hans Krebs in 1930. And basically what happens, it totally destroys acetyl-CoA. It removes the carbon and oxygens and throws them into the atmosphere as CO2, like just happened right here. It takes all the electrons and hydrogen ions away from acetyl-CoA, and it just totally destroys all of that molecule. So the glucose that you once started with is no longer the glucose that you end up with. You end up with absolutely nothing. It happens in the matrix of the mitochondria. So everything stays in the matrix. So again, Krebs cycle is also called the TCA cycle or the citric acid cycle. I don't know why. It just has all these different names. So now it's a cycle. It's in the shape of a circle. I'm sure I'll show you something at the end. I want you to get all the different reactions and what happens in the reactions and the enzymes that do the reactions, just like glycolysis before. You should be familiar with these things. So we have acetyl-CoA, and we're going to add it to oxaloacetate. And the reason I say 2 is because we have 2 pyruvate ending up or finishing glycolysis before, and 2 that enter oxidative decarboxylation, and 2 acetyl-CoA that exit. Now, we're going to see oxaloacetate again. This right here is recycled. This is totally destroyed. So acetyl-CoA is totally torn apart. So we have a 2-carbon molecule, acetyl-CoA, added to a 4-carbon oxaloacetate, and we get a 6-carbon citrate. And the enzyme is called citrate synthase. What we're doing is making citrate from adding acetyl-CoA to oxaloacetate. We're going to take the 2 molecules of citrate and we're going to form isocitrate. The enzyme is called aconitase. So we change it to an isomer of citrate. Why? Probably to reduce the potential energy of the bonds so that we can take away electrons easier. The 2 molecules of isocitrate are going to have a molecule of isocitrate is going to have 2 electrons and a hydrogen ion removed from it in the process. Carbon dioxide is released. There should be a plus CO2 down here. So 2 electrons and hydrogen ion are given to NAD to form NADH. There we go. We're going to get rid of a carbon dioxide there. So now we have a 5-carbon alpha ketoglutarate. Love that name too. That was like the second name. AKG would have been the second name beside PEP. The enzyme is isocitrate dehydrogenase. Why? Because we're removing 2 electrons and a hydrogen ion from isocitrate. That's the name. It's all in the name. The next step, which would be the 4th step in the series, we've taken alpha ketoglutarate. We're going to remove 2 electrons and a hydrogen ion from AKG here. We're going to give it to NAD. Right? We're going to add 2 molecules of coenzyme A. It's a different coenzyme A there. Okay, so well, we know that the removing of 2 electrons and a hydrogen ion are done by dehydrogenase. It happens to alpha ketoglutarate. So the enzyme is alpha ketoglutarate dehydrogenase. We form CO2, which goes into the atmosphere. Now, interestingly enough, there are no more carbons and oxygens left of that glucose molecule that you added. The only thing left are the hydrogens and electrons. That's it. So we have 2 electrons and a hydrogen ion added to NAD to form NADH. And we have the 4-carbon molecule called succinyl-CoA. Yep, I did say succinyl-CoA. All right, succinyl-CoA. And the enzyme is alpha ketoglutarate dehydrogenase. Well, so succinyl-CoA, we're going to form GTP from succinyl-CoA. The enzyme released when we remove the coenzyme there is able to add a phosphate to GDP. It's not gosh darn phosphate. It is guanosine diphosphate. We add a phosphate to that, we get GTP, which is guanosine triphosphate. It's the same thing as ATP. In fact, the phosphate from GTP is added to ADP to form ATP. When the GTP gives the phosphate to ADP, GTP goes back to GDP, and we start the whole process all over again. Isn't that crazy? We're moving phosphates, so we know that it has to be a kinase. See the kinase right there? So the enzyme is succinate thiokinase. The process, you form 2 succinates as well as 2 ATPs. And this coenzyme A is recycled to go back and add to alpha ketoglutarate in the previous step. I hope that made sense. It seems a little confusing. But we can go over it in class if you ask nicely. So 2 succinates, we're going to take 2 electrons and 2 hydrogen ions. This is a different electron carrier, FAD, flavin adenine dinucleotide. Flavin adenine dinucleotide. What was that? Flavin adenine dinucleotide. We're going to take 2 electrons and 2 hydrogen ions from succinate. We're going to add it to FAD, and we're going to get 2 molecules of fumarate and 2 FADH2s. They pretty much behave the same as NADHs. Just, I don't know why. That's the way it happens. We're removing electrons and hydrogen ions from succinate, so the enzyme must be, wait for it, succinate dehydrogenase. I know, I was so excited. We're almost there. We have now fumarate. We're going to add 2 other molecules to form malate. The enzyme is fumarase. That was a pretty easy step. The last step, we're going to take 2 malates. We're going to take 2 electrons and a hydrogen ion away from malate to form 2 NADHs, and we're going to form 2 molecules of oxaloacetate, which is where we started. Now, the enzyme that takes 2 electrons and a hydrogen ion away from malate is called malate dehydrogenase. What a surprise. So that is the Krebs cycle. That was done pretty quickly too, and hopefully you got it. If you need to listen to it again, listen to it again. And I bet I have the Krebs cycle right here. Number of carbons, the molecules here. So acetyl-CoA comes in here. Here, adds to oxaloacetate to form citrate. Go all the way down here. Alpha ketoglutarate, isn't it? Succinate, succinyl-CoA. Succinate, fumarate, malate, oxaloacetate. Beautiful. So that is the Krebs cycle. Yeah, it's a diagram. Pay attention to the steps. Yeah. So what do we have? We have a giant mess. We have totally destroyed the glucose. The glucose that came in has been totally broken up. The only thing left are the electrons that are carried and the hydrogen ions that are carried by the electron carrier. That's it. Electrons and hydrogens. The glycolysis, we end up with 2 ATP and 2 NADHs. Oxidative decarboxylation, we end up with 2 NADHs and 2 CO2s. Krebs cycle, we end up with 2 ATPs, 6 NADHs, 2 FADH2s, and 4 carbon dioxides. Okay, the CO2 goes into the atmosphere, probably goes to photosynthesis, which we talked about prior, so they can make oxygen so that we can do aerobic respiration. It's a giant cycle. It's so beautiful. The ATP, and we only have 4 ATP that are produced there, are used as energy. Good. Now, the big thing are the NADHs and the FADH2s. You notice we have 10 NADHs. We have 2 from glycolysis, 2 from oxidative decarboxylation, and 6 from the Krebs cycle. That's 10 of those, and we have 2 FADH2s. So where do they go? Well, they go to the electron transport chain. You're kidding me, right? No, the electron transport chain, and that is also known as the ETC. So they're on the inner membrane, protein chains, and they go in a specific order. Okay, so what they're going to do is they are going to pass electrons, just like photosynthesis, from protein to protein. So NADHs pass the 2 electrons, and we're just talking about electrons here, to flavin mononucleotide. Flavin mononucleotide passes the 2 electrons to an iron sulfur protein, that's FES, iron sulfur. FES passes the 2 electrons to ubiquinone, which is Q. That sounds like pastaquinone, I know. Q takes 2 hydrogen ions and 2 electrons and 2 hydrogen ions and pass the 2 hydrogen ions into the intermembrane space. Sounds just like PQH2. It is exactly the same thing, except it's moving the hydrogen ions from the matrix into the intermembrane space. 2 electrons are passed from QH2 to cytochrome B, which passes 2 electrons to cytochrome C1, also moves hydrogen ions from this matrix into the intermembrane space. C1 passes it to cytochrome C. Cytochrome C passes to cytochrome A. Cytochromes are all proteins. And cytochrome A passes to cytochrome A3, which also moves hydrogen ions from the matrix into the intermembrane space. The very last step is that cytochrome A3 passes 2 electrons to oxygen, which combines with hydrogen ions to form water. Now, in this step right here, you can form free radicals, the OH free radicals, hydroxyl free radicals, which can damage DNA. And that's where the superoxide dismutase comes into play to produce hydrogen peroxide, which is then broken down by catalase. The cyanide, if you take the poison, blocks this last step here. So electrons can't go past here. So basically the electron chain gets backed up. As the electron chain gets backed up, Krebs cycle gets backed up, and you stop producing energy. And if you can't produce enough energy, you will die. The FADH2, what happens to that? Well, they pass the electrons to Q instead of passing the electrons to FMN. That's all that does. The whole purpose is to increase hydrogen ion concentration of the intermembrane space. And you might be asking yourself, why does that happen? And in the next screencast, I will tell you why, and I bet you actually know why. It has to do with ATP synthase. So if you need to, and I know that was very quick and I apologize, but remember, just passing electrons from protein to protein and moving hydrogen ions from the matrix to the intermembrane space. That's all you're doing. And passing the electrons, the final electron acceptor is oxygen. That is the only reason you breathe, is so that you can have oxygen to accept those electrons from cytochrome A3. That's it. As long as you have enough oxygen to do that, you can produce energy and you will live. Reason you drown, you can't get the oxygen, low concentration of oxygen in the water. You can't get enough oxygen into your mitochondria to accept the electrons. Hope that helped. Hope that was interesting. I know it was fun for me. I'll talk to you in a bit.